Hello students, welcome to the Microbiology lab. Today's experiment is endospore staining. So as you know, like normal bacterial cells are vegetative cells and they can freely divide or multiply when a suitable condition is given, like the availability of the nutrients, when proper supply of oxygen, temperature, moisture, everything is given, they can divide rapidly. But in some species of bacteria, when they are exposed to unfavorable conditions such as lack of nutrients or limited supply of oxygen or carbon dioxide or limited supply of appropriate temperature, what they do is they form a protective layer around them, making them adapted to the unfavorable condition, and such structures are known as the endospores. So these endospores can revert or return back to the vegetative form when the favorable condition comes. And these endospores can be present in the end of the bacteria or the bacteria, or in the middle of the cell, or even between the end and the middle portion of the cell. And they can also exist in spherical or elliptical form. Endospore staining is a differential staining technique which is used to identify, detect and differentiate vegetative cells and endospores. So here we use Schaefer-Fulton's method of staining endospore, where the primary stain used is malachite green. Because the endospore resists the stain, what we have to do is we have to force a malachite green stain into the endospore with the process of heating. So here the heating process acts as a mordant. Then the malachite green is said to be soluble, and they have less affinity for the cellular materials, especially for the vegetative cells. So we can decolorize it with water. And these vegetative cells that has been decolorized by the water will be counterstained by the application of safranin. As a result, the vegetative cells appear to be pink due to the counterstained safranin, and the endospore stain means the endospore appears to be green color, bright green color, due to the malachite green staining. Let's see what are the requirements needed for the experiment endospore staining. First of all, we need the microscopic slide. The microscopic slide should be clean and neat. Then the primary stain malachite green, the counter stain safranin, and tap water or distilled water as the decolorizer. And we have the bacterial cell suspension here for making the smear, and a sterile loop to prepare the bacterial smear. The first step in the endospore staining is the preparation of the bacterial smear and heat fixing of the same. So for that we have to take a clean slide, microscopic slide, and the bacterial suspension, a sterile loop. With the help of a sterile loop, take your bacterial suspension, a loop full of bacterial suspension, and prepare a clean smear. Now, after the preparation of the smear, you have to heat fix it by passing your slide over the flame. Now we have heat fixed the bacterial smear. The next step in the procedure is we have to saturate the smear with the malachite green. For that, the heat fixed smear is taken and we have to saturate with malachite green. After saturating the stain with a slide, we have to keep the slide. We have to give steam for almost 5 to 10 minutes. So for that, we can place it over the slide and give the steam for almost 5 to 10 minutes, because this steaming acts as a mordant so that the malachite green will be forced into the endospore. This is the steamed malachite green stain over the smear. So after that we have to wash it with the water as a decolorizer. So kindly by tilting it, you have to wash it properly in order to decolorize it. So after the decolorization, we have to apply the saffron. Cover the smear with saffron. The application of the saffron, you have to wash off the stain with water. Excess stain should be washed off with water. Now you can view it under the microscope. So I already told you the vegetative cells appear to be pink color or kindish of dark brownish pink color, and the endospores appear to be bright green color. Now we will try to focus the slide under the microscope. Mixture of endospores and a bacterial cell, that is mostly saffron stain.